morning. I'm Amy Lensing-Tate from the UWM Alumni Association. Thank you for joining us for our new Mobile Master Chat series. While we can't be together on campus, we're happy to bring you weekly talks from our distinguished faculty and professors on a wide range of topics and disciplines. Today, I want to thank the UWM Graduate School and the George Kelps Lecture Fund for sponsoring this chat. It's my privilege to present Dr. Rebecca Klaper, Professor and Director of the Great Lakes Genomics Center at UWM School of Freshwater Sciences. Dr. Klaper and her lab conduct research to inform policy decisions involving freshwater resources. She studies the potential impact of emerging contaminants such as nanoparticles, pharmaceuticals, and compounds on aquatic life, and the impact these chemicals have on aquatic and human health. She's also part of an NSF-funded Center for Sustainable Nanotechnology. She's also the recipient of a Fulbright Award to the United Kingdom and a Science and Technology Policy Fellowship at the U.S. Environmental Protection Agency. With that, I'm happy to turn things over to Dr. Klaper.
Thank you so much. Thank you for, there it goes. Thanks so much for inviting me to give a talk. And thank you all for coming to listen too. As was said, I'm Rebecca Klaper, and I study the impacts of emerging contaminants in freshwater systems. I've been at the University of Wisconsin, Milwaukee for about, since 2003. So a fairly long time now. And one of the reasons I was attracted here was because of the Great Lakes system that's here right out our back door. Is such an important freshwater system. And so when I got here, I decided to look at these emerging contaminants as potential chemicals that would harm freshwater systems, and particularly the Great Lakes, and look at what kind of impacts they might have on the Great Lakes. Oops. There we go. So what are emerging contaminants? Sometimes they're called trace organics. And I'll get into that in a second, but there are things like pharmaceuticals, personal care products, brominated compounds like the flame retardants, they're often called PBDEs, fluorinated compounds that you're hearing a lot about in the news lately, PFAS compounds that were in firefighting foams and are also in nonstick kind of bakeware and cookware and in surfaces. Musks that go into our perfumes and shampoos and a lot of the things that you use to make your house smell nice. Things like phthalates that are in our plastics and bisphenol A that soften the plastic and allow it to be molded into particular shapes. Bisphenol A was in the news several years ago as they found that it is an endocrine disruptor, which I'll talk about in a little bit. And particularly they're worried about it in baby bottles. So you heard a lot about bisphenol A and B. And then these last two categories, pesticides and nanomaterials. So emerging contaminants are kind of, you can see there's a whole suite of different kinds of chemicals. Emerging contaminants is kind of a catch-all for all of these things. And they're divided up into kind of two groups. So you have new chemicals, things like nanomaterials, which are particulate chemicals that are down smaller than a red blood cell and have just come on the marketplace within the last 2 decades and are being put in lots of different things. They're really great properties that end up allowing us to do things like deliver chemotherapy agents, particularly to tumors, because they're so small and they can be targeted to particular areas in the body. They're in sunscreens because of their small size. They are very reflective as sunscreens. They're in all sorts of different things. Those new chemicals, and then and new pharmaceuticals, new pesticides on the market. And then we have a bunch of other things like pesticides that have been around for a really long time, pharmaceuticals that have been around for a really long time. And we're just within the last couple of decades able to detect them at the very low concentrations that they occur in the environment. And so the kind of those two different types of scenarios. And the main thing is that we are now able to detect these things all over the place, particularly in freshwater systems, but we're not really sure what any one of them does at the very low concentrations that we find them. So we're talking about nanogram per liter concentrations. If you think about a pharmaceutical, often we're taking those in thousands of milligram concentration. So the comparison is, they're very low concentrations. So the interest in these is because of that detection, but there's still a huge question about what kind of harm they might cause because they are at such low concentrations. Oops. These compounds have been found all over the United States. In fact, my interest in doing this kind of research came because of article by scientists at the U.S. Geological Survey that was published while I was finishing up graduate school. And it talked about how pharmaceuticals had been detected all over the United States in different freshwater systems. So looking at different streams in particular across the United States. And now we know that they occur in these freshwater systems, even more freshwater systems across the United States, around the world. And we also know that those same stream systems are impacted. So they show some evidence that the organisms that are in those particular water bodies are not doing so well. They're not thriving. They have some indications of things like endocrine disruption, changes in their hormones, and in their physical state because of the association with those sites. And the question is, well, are those pharmaceuticals, personal care products, those emerging contaminants, the cause of the changes that we see within these organisms? As I said, the question is, are they harmful? So we try to figure out which organisms, including humans, are exposed to the chemicals that are out in the environment and at what concentration. So you can detect these things all over the place, but are they at a concentration that you really see some kind of physiological impact on the organism that you're talking about? Is the chemical toxic at the dose that they're being exposed? And sometimes the answer is yes, and sometimes the answer is no. How do these things get into our water in the first place? Basically, so if you take a medication, your body basically is trying to eliminate that foreign chemical from itself. And so it will process it by sometimes breaking it down, but most often it goes right through you. Your kidneys and your liver, et cetera, do a really great job of getting rid of things in your body. And so you end up excreting quite a bit of the pharmaceuticals that you take into the sewer system through the toilet, and they go to our wastewater treatment plant. And then the wastewater treatment plant really wasn't designed to take these things out. It's really designed to get rid of bacteria, to process some of the organic material. I remember I called these things trace organics. These are a tiny amount of the organic material that are in those sewage treatment plants. And so the sewage treatment plant does take care of some of the pharmaceuticals and gets rid of them. But there's a small amount that goes out into the environment and to our freshwater system from there. And that's the amount that we care about. The flame retardants and some of the coatings end up washing off in the washing machine and go into the sewage treatment plant as well. We also use the biosolids from our treatment plants across the state and spread them onto agricultural fields, believe it or not. And so there's a chance that the stuff that ends up in the biosolids ends up getting spread into our agricultural systems, which is of concern, and then also wash into our freshwater systems from there. They're also used as fertilizers for parks and for sporting fields, et cetera. And finally, even though we have this great sewage treatment system, the piping and the infrastructure that we use to get the stuff to the sewage treatment plant is really old. And so we have leaks all over the system. And so some of the sewage that's being transported leaks right out into our groundwater systems, into surface water, into soils around neighborhoods. And so we end up having it enter our environment that way as well. Then the question is, who's exposed after it goes into a freshwater environment? So when I first got to the university, I grew up in Illinois, but love Wisconsin. I'm very glad to be in Wisconsin. But looking out the back door, I wanted to know what impact these things would have in the Great Lakes, and could we even detect them in such a huge water body? So a lot of the research has been done at sewage treatment plants that feed into river systems or small lakes, where it's very easy to track. Here's a source, this outfall of the treatment plant, and then going a certain distance downstream from that sewage treatment plant, you can detect usually less and less of the particular chemicals that are coming from the treatment plant. But if you have a system like Lake Michigan, it's like an ocean. And so what happens to those chemicals when they go out there? And, you know, the thought before is that, well, they come out at a very low concentration, and then you feed them into this huge water body that you probably wouldn't be able to detect much of anything because it would be diluted so much. There's a lot of mixing that goes on. And so our initial study was looking at, this is South Shore Treatment Plant that I have a picture of up here on the left, and looking at the input of the emerging contaminants, specifically mostly pharmaceuticals, going through the different stages of the wastewater treatment plant on several different days throughout the year, and then going out into Lake Michigan, and then following that the next day or so out in the lake, several miles out into the lake and to the south of the sewage treatment plant, and looking to see if we could really even detect any of the things that we knew were going in and being processed by the treatment plant. So overall, what we found is that we were able to measure a good number of pharmaceuticals and personal care products in the Great Lakes. And some of them were at levels that the Environmental Protection Agency, through their databases, said were of concern. And here's just an example. This is kind of a crazy graph, but you can see Lake Michigan. You can see South Shore Reclamation Facility, the wastewater treatment plant, which is down to the south of us, down near St. Francis and Oak Creek, and right on the shores of Lake Michigan. We also did some sampling up near Jones Island, and in that particular area, which was even more concentrated, as you can imagine, with the sea walls that are there. But in any case, we would detect the stuff coming through the South Shore treatment plant and then look at what happened going on in the lake. Each of these little boxes represents 25 nanograms per liter, again, very tiny amount. But if you can see that some of these things, they're actually very large concentrations comparatively. So we saw things like, and I've forgotten which, if it's the blue or the red, but I think it's the red, is caffeine coming out of our wastewater treatment plant. And this is often used as a marker. You know, a lot of people in the population drink their coffee and their soda, and we end up excreting a lot of caffeine going out into the sewage treatment plant, and it goes out into the lake at a certain concentration. I'll say that there's a lot of caffeine that goes into the treatment plant, and a good amount of it ends up getting removed. But the fact is, because there's so much coming in, the sewage treatment plant does not get rid of all of it, and so you see a good amount of it going through. And you can see that it goes several miles out into the lake. So the farthest one over to the right here was, I think, 3 kilometers out into the lake, so a few miles out into the lake. The biggest box here is just right off of the outfall of the sewage treatment plant. One of the other things to note, though, are these yellow boxes here. And this was a drug that I really hadn't thought very much about before. I was mainly interested because of what I had seen were the highest prescribed drugs, according to one of the pharmaceutical surveys, were the neuropharmaceuticals. And one of these small boxes that doesn't have very many colors here is fluoxetine, which is one of the main drugs that I had been studying, looking at effects, which is Prozac, and looking to see if that had any impact on fish. And I'll talk about that in a second. But these yellow boxes were the drug Metformin, which is a type 2 diabetes medication that I hadn't really ever heard of before, but it turns out it is prescribed at a very heavy amount in the population. And we could see this going on in the lake. It's not taken care of. It's not removed to a huge extent by the sewage treatment plant, but some of it is removed. But because, again, there's so much going in, we saw a lot coming out. And we could see spikes coming in and out of the sewage treatment plant, and then seeing those spikes going out in the lake. So overall, if you looked at and averaged the amount that we saw, we took about 6 samples throughout the year, because there is variation. You can imagine things like the antibiotics are taken in a more heavy clip in the wintertime when there are more infections and things going around. What we saw overall is an average of metformin that was fairly high, even higher than caffeine. And if we made an estimate that if you looked at the amount of discharge coming from that sewage treatment plant over the year, you'd end up seeing that metformin was being emitted about 14,000 pounds per year. So here, even though we think of little tiny amounts coming out each day at each hour, if you add it up over time, it's actually quite a bit of chemical coming out into the lake from that sewage treatment plant. And so in summary, we used to think of these things as minimal contributions, but collectively they might be significant. And some of them are still pretty low, 160 pounds per year for ampicillin, for example. Oh, I'll say too, I didn't describe what some of these other things are. I'm sure people are familiar with caffeine. Acetaminophen is pain reliever, Tylenol, naproxen, again, another pain reliever. So you can see things that people take quite often. Triclosan is an antiseptic that was put into hand soaps. It's in some toothpaste and other things, and it's more recently been banned because they were finding it everywhere because of those antibacterial hand soaps. And it turns out to be quite a contaminant in the environment that ends up having some biological effects. Let's see, it's thinking about it. There we go. So previous arguments about these chemicals, you know, we weren't too concerned about them because they come out on an hourly or daily basis at pretty low concentrations. Again, those nanogram per liter concentrations. And for pharmaceuticals, it doesn't come anywhere close to being a therapeutic dose for a human for pharmaceuticals. And so the argument was that they're not reaching toxic levels. And the other thought was that they would break down in the environment. So environment's kind of a harsh place for a chemical floating around. And so the thought was that they would break down. However, what we know is that, you know, pharmaceuticals are designed to go through your digestive system. And so they're fairly resistant to breaking down in that acidic environment, and also fairly resistant to breaking down in the environment. Bacteria and other things can break down some of the chemicals. UV light from sunlight ends up breaking down some of the chemicals, but not all of them get broken down. That metformin is a prime example of one that is not easily changed into anything else. We also know now too that low doses of pharmaceuticals can also push those same pathways in organisms. So for example, you might take thousands of milligrams of a particular chemical, but even if you took a little bit, it's still going to do some kind of chemical interaction with the pathways that it's designed to interact with within you and within other organisms. And lastly, if you think of the body size and the physiology of some of the organisms out in the environment, they're much smaller than us. And so the dose ends up being higher because of that body size. And then also because some of the organisms in the environment are physiologically different than us. And so, for instance, fish sometimes will take up chemicals at a greater rate than humans will into their tissues. And so it can affect the fish, for example, at a much lower dose than it would affect a human. So about the time that I was studying these, you know, word had gotten out that hormones that were coming out, like birth control pill medications, could be having hormonal effects on fish and other things, because that was the first thing people studied. They saw changes in fish populations, and the first thought was, well, it's hormones. So what hormones are coming through the treatment plant? Well, birth control pill medications come through the sewage treatment plant. You'll notice on my list, though, I didn't have any kind of chemical that was related to birth control pills on that list of chemicals. You didn't see an estrogen or progesterone, and that's because they come out at really tiny amounts. We're talking 4 nanograms per liter or less, which is really up until the last, I don't know, 7 or 8 years was the detection limit of our instruments. And so we couldn't even measure really the concentrations down that low. And yet we knew that tiny concentrations of hormones are bioactive in fish and other things. But so the Wisconsin DNR has a unit that's located in our building, and of course we're friends with those folks, and they talk to us about what's going on with the fish populations out in the lake, and particularly perch. And there was one of the scientists approached me and said, hey, you're studying those hormones coming out of the sewage treatment plant. We're seeing a huge decline in perch, and it's been attributed to harvest, but we're seeing these weird physiological things with the fish, which can't be, you know, associated with the gill netting of these fish. And so here you see a graph on the bottom of the drop and catch that's happened over the last several decades in perch. And we know that's for a few reasons. It is some overfishing, also the change in the food web structure of the Great Lakes because of the invasion of zebra mussels and quagga mussels that have eaten up the bottom of the food chain. But that doesn't necessarily explain some of the things they were seeing, which were these weird structures in males. So on the top right, you can see a male perch with a lovely testes organ that's all milky looking and fat. And this is a normal male perch going through reproduction. On the bottom, what you see is another male perch, but the testes are just really weird. They're very opaque and they don't have sperm in them, and the fish kind of looked weird. And his question was, well, could the hormones that are coming out of the sewage treatment plant end up causing this change in the fish? And so we set out to study this, and we collected some perch. And of course, the year that we went to go collect with him, when he first described this of what they were seeing, we couldn't find any more males. And so the males had disappeared, which is really weird. And so we caught a bunch of females, and we saw some differences in the females, but they're pretty minimal. And it really seems that the males are the ones that are changing, or at least that year that he found this weirdness in the testes, that the males were the ones that were bearing the brunt of whatever was going on. So we couldn't study the perch out in the lake for this. And so what we decided to do was do a laboratory-based study. And we decided to start with the chemicals that we found coming out in the highest amount. So we started with metformin, of course. We found that it was coming in at a fairly high rate of 100 micrograms per liter. So that's pretty high compared to any of the other chemicals, any of the personal care products and pharmaceuticals, I should say. There are other chemicals that come in at a higher clip. And it was going out at 47 micrograms per liter, which is really high compared to what anybody else had documented. But we also have an urban population. Here's some example of some of the other countries. We have a pretty urban population, a high number of people that are diabetic or pre-diabetic in our population. And so it might not be surprising that we had a higher concentration. I will say that there were other studies that came out just before and after ours that showed, for instance, there are several locations in Indiana that had concentrations that were around 40 micrograms per liter. So we aren't alone in this. So initially we started looking at metformin and thinking of it as being a metabolic problem. So people take metformin for insulin resistance and for insulin problems. And so we looked at the physiology of the fish in relationship to its metabolism, and we just really didn't find anything. But when the DNR came and talked to us about what was going on with the perch in Lake Michigan, we started wondering if there were any of the compounds that would be related to the endocrine system. So the things that create hormones that affect the sexual organs of the fish. When we look at metformin, it's kind of a weird molecule. It's got all these nitrogens and amide groups, and it doesn't look like a hormone, which has a more, here, let's see. It doesn't resemble the known endocrine disruptors. So if you look at some of the hormone active agents, they have these ring structures. And so you can see those two molecules don't look anything alike. However, one of the things that we found out from our literature searches is that metformin was starting to be prescribed for this condition called polycystic ovarian syndrome, which some of you might have heard of, or PCOS, where females have lower progesterone levels, and it ends up affecting all sorts of things. PCOS initially was identified as people that are insulin resistant, and particularly women, and that have hormone problems, irregular periods. They have hirsutism, where you grow a hair on your face at a thicker and faster rate. It is associated, though, also with these changing hormone levels, and people with PCOS often have problems with fertility. What happens is you create too many eggs at once, or follicles start maturing at once. And normally, if you remember way back to your high school biology, what happens when a woman goes through her menstrual cycle, what's going on is that one follicle develops over time and develops into a little egg, and the egg is released and then gets fertilized, etc., or does not. But what happens in a woman that has PCOS is that there are a bunch of follicles all developing at the same time. And so she has fertility problems because none of them are really of any great quality. And so it prevents her from going through with pregnancy. And so the question is, what happens in fish when they're exposed to this chemical? I'll say that, I know this, you're looking probably at the headline right now, but when we exposed adult fish to this chemical at the rate that was coming out in the environment, we really didn't see much of a change. What we did see is some changes in the gene expression associated with hormones in male fish, and really nothing in females. But then we started realizing that if we were going to catch what was going on with these fish, we had to look at probably what was going on from their birth all the way up through adulthood. And because that's when the sexual organs would be developing, and you'd most likely see the impacts of the chemical that would be associated with what the DNR was seeing out in the field. And so we took these little fish, fathead minnows, these are native fish in Wisconsin. They are often used, if you go into a bait shop, you can find these guys. They are used for musky fishing and big fish fishing. But they're cute little minnows that are found in our freshwater systems. They mate by, the male makes a nest and then uses that big fat pad on the top of his head. The male is the one with the fat head. And I'll let you come to your own conclusions about that. But he uses that fat pad on the top of his head to clean the nest, which is usually a branch or something or a rock in the stream, and then attracts the female and they mate. And then the male takes care of the eggs, and then over time, when they hatch, he lets them go. But the ones that are not hatching and have problems, he'll pick out the fungus and the bacteria and stuff that's growing on the eggs. It's really lovely organism to look at reproductive changes. But one of the things that we saw is by exposing these guys to that 40 micrograms per liter of the metformin, the males developed this intersex condition where the males, you can see on the top right, you have these kind of purplish pictures. And on the left is a normal male. So you have the testes have some of these little pockets in them, and they're, but this is by and large what the testes look like. On the right-hand side is the most extreme condition that we saw in the metformin exposed males, where we saw these round structures developing, these ovotestes basically. So where are these egg structures developing within the testes of the male fish? So, and it was very dramatic change in the exposed males. We also saw that when you paired up these male and female fish that had been exposed, you saw a decline in the number of clutches of eggs that they laid. So the number of times that they laid eggs in these metformin exposed fish. And so we saw this change in reproduction that was going on. We also saw that the males and females were smaller that were exposed to the metformin. So there is some kind of physiological change going on, but the most dramatic thing was this intersex condition. And right now what we're trying to do is figure out what's going on in the fish. Trying to get some funding to look and see what pathway is really being triggered. It's really strange. Metformin, I'm sure the pharmaceutical companies know more, but it's still unclear as to what exactly is going on with metformin in humans, let alone in little fish, that might end up causing something like this. And so what we're doing now is we're looking to see what's going on with physiologically in the organism using genomics and other technologies to look at this. So what about some of the other chemicals? Oh, thanks. So one of the things that we're doing also is looking at, there's a whole suite of chemicals that come out of sewage treatment plant. We try looking at them individually, but then also collectively and mixtures, because it's not just one chemical coming out, it's a bunch of them, to look and see what kind of impacts overall that effluent of emerging contaminants might have on organisms. So let's see, just taking a second. So one of the compounds that I started out with, I mentioned, was fluoxetine, because there had already been documented that that was coming out in sewage treatment plants. And Prozac, back when I first started, was listed as one of the #1 chemicals that people were taking, pharmaceuticals that people were taking therapeutically. And so what we did is one of my students and I set up an experiment looking at fluoxetine and its impact on behavior and reproduction in these same little fathead minnows. You can see the little nest we made up here on the right-hand side for the fathead minnows, and you can see the little male in there cleaning away at the eggs that are stuck to the top of that PVC pipe. And we chose, for each of these chemicals, we try to choose, since the pharmaceuticals are designed to work in a certain way, we try to look at the mechanisms that are supposed to be triggered by that chemical when a human or another organism is exposed to them. So for fluoxetine, what we did is we looked at behavior. You have Prozac, it's supposed to be a behavioral drug. We looked at behavior and also looked, because we care about populations of critters out in the wild, we looked at how it affected reproduction, the population numbers of those animals. Overall, what we saw is that males spend more time under the mating tile to the detriment of going out and finding a mate, and that it took a longer time for them to go out and capture prey. And in the most extreme conditions of 100 nanograms per liter, you end up seeing that the males end up attacking the females and killing them, actually, which is not a great response. The good news is, though, is that we often find fluoxetine down at 10 nanograms per liter and below. Or I'm sorry, 100 nanograms per liter and low. And this is up in the microgram per liter concentration that we were seeing these effect. And so chances are that this alone is not necessarily causing a change in these fish out in the wild. One of the key things that we don't know about are the effects of mixtures of these chemicals, and how different chemicals that act on the same pathway that are out in the environment might collectively end up impacting critters. And so this is an example of a paper by my former postdoc and myself, looking at linuron, which is a pesticide, and DEHP as a plasticizer. The overall impact of this graph over here down on the bottom right is showing that each of these is said to be an endocrine disruptor, but when you look at the concentration that they're found out in the environment, those second and third columns indicate that they're not really different than the control. So they're not really causing any kind of endocrine disrupting effect at the concentrations that you find them in the environment. However, when you mix the two of them together, it ends up decreasing the amount of testosterone that a fish produces. And so collectively, they might be causing a greater impact than either one of them by themselves. So just going back to that fluoxetine, there are other chemicals that interact with the nervous system of vertebrates that are being released in the environment. So it's possible that fluoxetine plus any of the other neuropharmaceuticals that are out there could collectively cause a bigger impact than any one of them by themselves. This is kind of a crazy chart, but the overall thing to look at is the fact that what we did is we looked at each of these particular chemicals alone at the concentrations that they're found out in the environment. So we do these dose responses going from very low doses all the way up to very high doses. And the yellow boxes represent the concentrations that we measured in Lake Michigan or Milwaukee River right at the drainage. The red numbers represent the cases where we saw a significant difference in this particular gene expression that's related to endocrine disruption, related to the control. And you can see that not all of these chemicals end up having an effect alone at the concentrations you find in the environment. And there are only certain chemicals that we really need to pay very close attention to. In this case, it's that Metformin, it's a transformation product, guanyl urea, and then the plasticizer DEHP. And so not all chemicals seem to have a significant environmental impact, but there are some that we really need to watch. So some of the questions I often get are about, well, what policies or regulations exist for these chemicals in the environment and what's needed? And we do have some things that regulate the chemicals going out in the environment that relate to these emerging contaminants. The Food and Drug Administration has set a 1 part per billion limit for environmental levels of these chemicals going out before they'll regulate them. The problem we're seeing, though, is that some of the effects are at the concentrations that are below that. And so the scientific community has called for reevaluating things that, especially that are hormone related. The Safe Drinking Water Act does also protect human health. A lot of these contaminants are monitored when they're flagged as being something of interest. So for instance, if you go to the Milwaukee Water Department's website, you can see all the different chemicals that they have monitored coming in to our drinking water pipes. And you find that most of them are at no detect levels. They're really low concentrations that you can't even detect with our instruments. And so we aren't necessarily getting exposed to those. And then there are also several other federal legislations that protect us against, for instance, pesticides that might be harmful out in the environment. However, there are some other removal tactics that we've been looking at, new removal technologies that might remove more of these things from the sewage treatment plant and whether they're worth putting in. Green chemistry, there are several companies that are trying to design chemicals that break down faster, that have less of a chance of interacting with organisms negatively out in the environment. This is a big movement in the chemistry and pharmaceutical industry. And then also changing our own habits about how many pharmaceuticals we really take. The United States is a great market for the pharmaceutical industry. We take a lot of medications. It's surprising when you go into a doctor's office and someone's not taking some sort of medication. And the question is, do we all need to be taking all of these medications? Obviously, some of them are really helpful, but could we be taking them at a lower dose? Are all of them really necessary? And so this is where my lab, this is where I'll end my talk, but this is where my lab is focusing right now, is what are these different places that we could change on this pathway in order to have the greatest environmental impact? Is it changing the way we design our medications and our personal care products, and really identifying during the trial and approval process which ones are harmful in different ways than what we're doing right now? Can we change our prescription use of some of these pharmaceuticals? Some of them have already been identified as being prescribed at really high concentrations maybe the way to deal with this is to decrease the concentrations in use. Of course, that's only for certain medications. It's not that everybody should stop taking their medications right now, but also changes in lifestyle so that we don't necessarily need to use so many chemicals in our bodies in order to function. And then also the end of life. What do we do at the end with the medications we haven't used? At the end of the waste stream of the pharmaceutical companies so that we can decrease how much is going out in the environment. And so my lab is now working on where along this pathway could we make the biggest difference. Lastly, I'll just say that it's not just me that is doing all this work. I have fantastic students and postdocs that come to me at University of Wisconsin, Milwaukee, that are participating in this research and being the leaders in the field and leading this research within my lab. And I can't do this without them. So thank you very much. And I'd be happy to take some questions. The questions I get is what do we do with our old medications when we have them? And does that make a huge contribution to what goes out into the environment? And I'll say that there are take-back events, and now they're very prevalent. There's the Take Back My Meds Milwaukee that's made a huge push to get boxes into pharmacies and in the police stations so that you can drop off your old medications. And what happens to those? Generally, they get incinerated. And so it's a different output that I don't know if anybody's really totally investigated, but it doesn't go directly into the to the water environment from there. It is definitely something to do with your old medications. However, the environmental community by and large thinks that most of the medications that go out in the environment are actually due to people taking their medication. And so the question then is, how do we tackle that? So one of the questions from the audience is there any research about thyroid medication? That's a really good question. So the US Environmental Protection Agency and the National Institute of Health has developed this screening going back to all the chemicals that hadn't been evaluated before. And one of the things they've been focusing on mostly is the estrogenic pathways and looking to see if there's any endocrine disruption from estrogens. And there wasn't really a great way because we know that the thyroid is a really sensitive place for chemicals. There wasn't really a great way to test for disruption of thyroid pathways. And so they've started developing some high throughput assays where we can look at that question a little bit more. And so there is some indication, for example, that some chemicals are thyroid active, and they've done a lot of that research on frogs, for example. It's much easier to see changes in thyroid pathway disruption in frogs as a model organism. And yes, there are some thyroid medications at lower doses that end up affecting thyroid and other vertebrates like frogs, for example. But you'll see probably more research coming out about that particular pathway and impacts of not only thyroid medications, but other chemicals that might impact the thyroid, things like pesticides and fungicides, for example, in the near future, I think. Yeah, how does this impact Milwaukee's drinking water? So as I said, Milwaukee water treatment plant does monitor for some of these contaminants of concern. And this goes past just pharmaceuticals, but also other emerging contaminants. And this has been for a while. So this is an EPA Safe Drinking Water Act push for looking at contaminants that are new or that we have new concerns about. Milwaukee's drinking water, since the cryptosporidium outbreak, has been moved. So it's farther away from the sewage treatment plant. It comes from farther out in the lake. And we don't see a ton of things coming into the drinking water system and very little coming out the other side from these emerging contaminants. And of course, we have problems with our distribution system with lead, et cetera. But for Milwaukee, I'm very glad that I live on Lake Michigan and that we're drinking Lake Michigan water, let's just say. It's one of the reasons that I live here, is because of our water source is pretty great. If you talk about people that are drawing water from downstream of a sewage treatment plant. And you can think of some river systems like the Mississippi, where it goes through, I don't know how many sewage treatment plants going from the top to the bottom of the Mississippi. You probably have a higher concentration coming in your drinking water treatment plant than we do. Also, there's a big push in arid regions where there's very little water, like Los Angeles, for example, and in Arizona, for recycling our water, going right from the sewage treatment plant effluent into a drinking water treatment system. Because they just have very little choice for a water source. And there is some concern there that there's not a great way to get rid of everything that's coming through that water treatment system and into a drinking water system. So it really varies by location in the country. But Milwaukee, I like. That's a good question. How many sewage treatment plants discharge water into Lake Michigan? And you know what? I don't know if I know the answer to that. It's definitely more concentrated at the cities that are going around the lake. So you have some small treatment plants going around the lake, and that's why I can't really totally answer that question. In the Milwaukee area, we have Oak Creek, that South Shore treatment plant. We have Jones Island. Down towards Chicago, you know, they've reversed the flow of the Chicago River, so it goes the other direction. And so Chicago really is inputting into the Mississippi. But there are suburbs around the Chicago area that discharge right into the lake as well. So in the northern suburbs of Chicago, going down to Gary, and then around over to Michigan, there are some sewage treatment plants that empty out in the lake. You see the FDA taking a stronger stand on the amount of extra medication people are taking and relieving. I don't see this happening, although it is a discussion in the scientific community that we're trying to push. And you know, there are great forces on the other side. Obviously, this is a capitalist society with a lot of pharmaceutical companies that have an interest in increasing the amount of medications that people are taking for a business model. And so I don't necessarily see that push, although you do see it from certain doctors that are familiar with this as a problem. And that's where we're trying to have conversations about this. Is there a way to decrease the amount of medications people are taking in order to help with this issue? And I think it's a really important discussion, not just for our water quality, but also for our human population as a whole, where, you know, are we really, is it really necessary for us to be taking all of the medications that we're taking as a population? You see huge differences between the United States and other countries in that regard.
Thank you very much, Dr. Klaper. Please join us next week when we'll feature Dr. Margot Anderson, a UWM Distinguished Professor Emerita and a national expert on the U.S. Census. She'll explain why the U.S. has taken a census every 10 years since 1790 and discuss the implications of the 2020 census being conducted at the height of the COVID-19 pandemic. Remember, if you can't participate live, these chats are always available on demand as well at the alumni website. Thank you again, Rebecca, for that fascinating talk today, and we hope to have you back on campus some point soon.
Me too, yes, and thank you all, and thank you, the Alumni Association, for inviting me.